Welcome to this explainer. Today we're diving into some absolutely fascinating genetic data to ask a pretty wild question. Can a biological mutation actually make you superhuman? It's a concept that sounds straight out of a comic book, right? But the human genome is vast, ancient, and full of incredible secrets. So today we're going to unpack the science behind one of the most remarkable genetic anomalies ever discovered.
Let's kick things off by looking at a literal, real-life superpower. When we think of superhuman traits, we usually imagine extreme physical strength. Well, in the realm of genetics, there's a rare condition known as myostatin-related muscle hypertrophy, and it perfectly sets the stage for our central question. How much can a single tweak in our DNA change what our bodies are actually capable of? Just look at this contrast. It shows how one single genetic tweak can literally double a person's muscle mass and increase their strength, all while reducing body fat. And the data shows this happens without causing any known intellectual or medical issues. Affected individuals are perfectly healthy, just remarkably strong. But while this visible physical strength is fascinating, our true detective story today lies with a much more hidden power. We're going to look at an invisible cellular superpower that completely changed the course of modern medicine.
Section 1. The Magic HIV Shield. So let's transition from physical muscle to an invisible cellular shield. Tracing the data back to the 1980s, we see the AIDS epidemic first emerged in medical reports in 1981. By 1984, the HIV-1 virus was discovered as the culprit. For years, this was just a relentless, fatal disease with absolutely no cure. But then in 1996, 11 years into this devastating epidemic, researchers uncovered a glimmer of hope hidden right inside human DNA. They were hunting for genetic factors that influence the disease, and what they found was truly astonishing.
What researchers found was this simple 32 base pair deletion, which scientists named CCR5 delta 32. It essentially removes the very doorway that HIV needs to infect our immune cells. You see, for HIV to replicate, it has to break into our CD4 T lymphocytes. These are basically the generals of our immune system. The virus does this by latching onto specific receptors on the cell surface, primarily one called CCR5. But this specific genetic mutation deletes just enough of the code to completely ruin that receptor.
Think of it like this. While a normal cell leaves the front door wide open for the virus, the delta 32 mutation completely removes the handle. The virus is locked out. The mutation produces a truncated protein that the cell simply destroys, meaning people with this mutation have absolutely zero CCR5 receptors on their T cells.
And guys, this isn't just a slight resistance, it is absolute. Out of nearly 20,000 infected patients studied by genetic epidemiologists, scientists found exactly 0 who were homozygous for this specific mutation, meaning 0 infected people had inherited the mutated gene from both parents. This proved that carrying 2 copies of the CCR5 delta 32 mutation acts as a near-perfect biological shield against HIV infection, completely regardless of exposure.
Section 2. Forged by the Black Death.
But wait, how did a shield against a modern virus get here in the first place? HIV is way too new for natural selection to have spread this variant so widely. Something else, something historical and massive, had to have forced this mutation into the population. The epidemiological data points to a fascinating geographic puzzle. We see a distinct gradient, with the variant appearing in 10 to 15% of Northern Europeans, like in Scotland and Scandinavia. It drops to 4 to 6% in Southern Europe and is virtually non-existent in Asian and African populations. In evolutionary biology, a distribution like this points to an immense, localized, historical selective pressure. Something deadly swept through Europe and spared those who carry this specific shield. Well, by analyzing chromosome lengths and genetic markers, geneticists calculated a sort of biological clock. They determined that the massive selective pressure causing this mutation to spike occurred about 682 years ago. That calculation points directly to the year 1347.
And that timeline? It perfectly aligns with the Black Death. This was a brutal cataclysm that wiped out up to 40 million Europeans in just 5 years, between 1347 and 1352. The bubonic plague decimated cities and the countryside alike. In genetic terms, this means those with a CCR5 mutation had a massive survival advantage. While millions perished, those with the missing cellular doorway were significantly more likely to live and pass on their genes. There's a wild irony to that, isn't there? One of the most horrific plagues in human history effectively forged the armor some people use to fight off a completely unrelated modern virus today.
And lab data actually backs up this historical detective work. Back in 2004, researchers tested the plague bacteria on mice. When they used knockout mice, which are engineered to completely lack the CCR5 receptor to simulate the human mutation, the cellular uptake of the plague bacteria by the immune system was reduced by an astonishing 30 times compared to normal mice. So the same doorway HIV uses today was the doorway the plague used centuries ago.
Section 3. Curing the incurable.
Knowing this mutation completely blocked HIV, doctors naturally started wondering, could we transfer this genetic superpower to someone who's already sick? Enter clinical hematologist Dr. Jero Hutter in 2006. He pioneered a radical treatment for an HIV-positive patient who also had a lethal case of leukemia. Dr. Hutter designed a protocol to treat both at the same time. First, he irradiated the patient's failing immune system. Then, he scoured registries to find a bone marrow donor who was homozygous for the double CCR5 delta 32 mutation. Finally, he transplanted these superhuman HIV-resistant stem cells into the patient.
And against all odds, it worked. The transplanted cells repopulated his body. The patient, Timothy Bray Brown, known globally as the Berlin patient, was taken off his antiretroviral drugs, and the virus never returned. He became the very first person in history to be officially cured of HIV. This unbelievable success proved the concept and actually led to the successful cure of 4 other patients using the exact same method.
Section 4. The Gene Editing Scandal.
Of course, the temptation to artificially engineer this magic gene was huge. In 2018, the scientific community was absolutely horrified when Chinese scientist He Jiankui announced he had used CRISPR technology to permanently knock out the CCR5 gene in human embryos. He implanted these edited embryos, which resulted in the birth of twin girls, known by the pseudonyms Lulu and Nana. His goal was to make them immune to HIV since their father was infected.
This unauthorized germline editing sparked massive global outrage. Why? Because altering germline DNA means those changes and any unforeseen side effects are permanent for all future generations. It brought swift legal consequences. Chinese courts convicted him with illegal medical practices, sentencing him to 3 years in prison and a $500,000 fine. It really highlighted the extreme dangers and incredibly complex realities of meddling with human DNA.
Section 5. The Superpower Trade-offs.
Which brings us to a crucial biological reality. In nature's design, there are 0 superpowers without a severe catch. The human body is a delicate balance, and removing a piece of our genetic code usually comes with a hefty price tag. We have to ask the data. What is the actual biological cost of permanently locking this cellular doorway? Because we know the CCR5 receptor wasn't just put there to give viruses a way in. It's a functional part of our immune system, regulating inflammation and fighting off other specific bugs. The dark side of this mutation is actually pretty stark. While it blocks HIV and the plague, it turns out you desperately need that exact same receptor to fight off other diseases. When scientists infected CCR5 knockout mice with the West Nile virus, the results were devastating. The mice without the receptor rapidly developed fatal encephalitis, dying in just 12 days. Meanwhile, 60% of normal mice with their CCR5 receptors intact easily survived the infection.
And tragically, that lab data translates directly to humans. In studies of people infected with West Nile virus, those carrying the double mutation make up a staggering 25% of the fatal encephalitis cases. Contrast that with just 8% of the mild cases. This proves unequivocally that this genetic shield is a double-edged sword. It leaves you immensely vulnerable, even fatally so, to other pathogens.
So if there's one thing to take away from all this data, it's that our genetic code is an ancient, heavily negotiated treaty with nature. Every biological defense carries a deeply hidden risk. Which leaves us with a final provocative thought to end our explainer today. Knowing what we know now about the delicate balance of our immune system, if you had the CRISPR tools right in front of you to rewrite your own DNA, would the superhuman benefits of total viral immunity truly outweigh the invisible, deadly risks?